I did so. I've not submitted my slide though because I couldn't complete it because of network issues. I chatted with someone yesterday, so I don't have what to bring.
Okay, there's no problem. The defense is open to everyone.
Yeah, thank you.
Others can listen and learn from those who did something. So I really want to appreciate everyone here. Dejote, Diola, Mr. Dr. Joseph, I can see Emmanuel Atintokun, I can see Salusi and Beneme Azota Odrechi. So we are all welcome. Um, it has been a wonderful experience. I really want to appreciate us who were able to do something. Reason is this: we use this medium, this mini project medium, to assess our performance, to see if we really have few persons who understands what we are doing, and few persons who can do something from what we have given to them at a given period of time. Yes, we have also had a lot of cases where some persons wish to participate in the mini project, but time and chance wouldn't have allowed them. Some have taken permission to be doing it whenever they are done. So I told them, no problem, till whenever we are done. And that will be probably the eternity. Because time and chance will never be there for any time. You will create the time and chance for yourself. So once again, thank you for pulling it, for making it through this moment. We are going to start as it is in the list, from the list in the platform. So who is number one from that list? Okay, let me quickly get the list. So we'll start. Okay, all right. I have Daniel, number one. So do you have Daniel here?
Yes, sir.
All right. So can you quickly share your screen?
Okay, sir.
All right.
Can you see my screen now, sir? Hello?
Yes, we can see your screen.
Yes.
Maybe you click show my screen anyway, so that lettering masking your screen will be off. There's your screen.
All right, thank you. Okay.
All right.
Can I start now, please?
Yeah, go ahead. You have the platform.
Okay, sir. Good evening, everyone. Welcome to the mini project defense for molecular modeling. Right. So, like we're all given an assignment, I chose to work with ibuprofen derivatives. Right. So I'll be talking on the molecular modeling aspect of ibuprofen derivatives. Okay. So, way of introduction. Molecular modeling refers to the use of computational methods and theoretical framework to represent, simulate, and predict the structures, properties, and activities of molecules. And of course, we all know that it has become an indispensable tool in modern drug discovery. And it's a very important step that must be taken before, proceeding for molecular docking. It's very vital that we do this first before proceeding for molecular docking. So in this work, about 4,000 derivatives of ibuprofen were screened, right? Actually, when I downloaded the derivatives from PubChem, I got about 8,000 derivatives, but I chose to screen 4,000. The assignment said we should do at least 2,000, so I decided to do 4,000. So I screened 4,000 derivatives. As well, no, ibuprofen is an NSAID, right? Non-steroidal anti-inflammatory drug used in the management of pain, inflammation, and so forth. All right. Like I said, 8,000 derivatives were downloaded from PubChem, right? So with the use of OpenBabel, those structures, right, that are downloaded from PubChem, the structures, with the aid of OpenBabel, I converted those structures to that my code. Then after doing that, the properties of those various compounds, of each compounds, was downloaded from AdmetLab. I use AdmetLab to, get the physicochemical properties. Okay. Okay. This is the okay. Okay, this is the smart code converted with OpenBabel. Then the admit feature I used, it was toxicological screening, so like what was explained to us in the class. So I decided to follow the same format. So I did what's it called, the carcinogenicity, neurotoxicity, autotoxicity, hematotoxicity, nephrotoxicity, genotoxicity, and of course the F50, the oral bioavailability, and of course the Lipinski rule of 5. So those were the parameters I used for this screening. Okay. Okay. Come in. Okay. The benchmark. Come in. The benchmark that I used was less than 0.5 for all of them. Less than 0.5. From 0 to 0.3 is a low risk. From 0.31 to 0.5, moderate risk. Then from 0.51 to 1, it's very high risk. But I just to go for the middle, 0.5. I use less than 0.5, right? So after doing that, out of the 4,000 compounds, right, that I screened, the Lipinski, the oral bioavailability, the genotoxicity, autotoxicity, and all of that. Out of the 4,000 compounds that I screened using a benchmark of less than 0.5, the number of sources I got was okay. Is it? I got 164 compounds that passed that benchmark, right? So these are some of the the results, some of those compounds that passed. 7-phenyl ethanoic acid, 2,2-diphenylbutyric acid. There are lots of I couldn't download, like I have to go back to PubChem using the smart code to download the structures. So I just downloaded it like a few, 15 or so. So I couldn't get the 100 and the structure of the 164 that passed, right? So these are the results. Okay, then okay, that is it. So in conclusion, we all know that high throughput screening is a process, in which large libraries of chemical compounds, in case of ibuprofen derivatives, 8,000 compounds were screened. So they are rapidly tested in order to identify molecules that show activity against a biological target. So out of the 4,000 compounds that were screened, only about 165 molecules passed the benchmark of less than 0.5 that was set. So it therefore means that we cannot proceed with this, this molecule for docking. So recommendations: molecular modeling, when combined with machine learning and AI, is a very helpful tool that can best predict the activities of molecules based on the references that we have set. And with integration of in silico toxicological prediction, researchers can systematically, identify safe and effective bioactive compounds. As more people begin to adopt these tools, there will be a rise, in the development of drug molecules that show specificity towards a biological target. That brings me to the end of my presentation.
Well, that was a good one.
Thank
So
you,
I
sir.
will, yeah, I will allow participants who have questions to ask. Is there anyone with question for him? Okay. Uh, what prompted the use of ibuprofen? Why did you choose to work with ibuprofen? Yeah, then can you hear me?
I can hear you, sir.
So why did you choose to work with ibuprofen?
Um, okay. Like we all know, ibuprofen is an NSAID. We all know that NSAIDs have this, GI effects. It has an effect that it can cause meningitis, this ulcer, you know, aggravating already existing ulcer. So I was just thinking, I was like, okay, if we get other, derivatives and see if we can, maybe modify. Though modification was not, I didn't do any lead optimization, right? But I was just thinking, okay, maybe we could get other derivatives. We could, um, optimize and see if we get a better derivative that is gastro friendly, that doesn't, uh, have that tendency to cause stuff like that. That's why.
Okay, okay, okay. That is great. And what was the choice? You worked on you screened based on toxicity.
Yes, sir.
So yes. So does it mean that there is a report or there is a case that ibuprofen already is toxic? Then you are looking for a better compound similar to ibuprofen that made you to actually look at that toxicity aspect, or you just choose it for the sake of the assignment?
I just chose it for the sake of the assignment.
Okay,
Yes, sir.
all right. You have done a very good job, and the results. I would have been grateful. Okay, the result was really really good, and just to make it a bit better, I think when we are doing further screening, we also screen with our heat, so that will serve as control, or yes, that will serve as control. So imagine if you have generated 4,000 ibuprofen derivatives, and you also included ibuprofen as part of the whole compound. So you screened. You may not see that some of these derivatives now may even have better properties than the ibuprofen, because that is our goal. Uh,
That's true, sir.
yes, that is our goal. But when ibuprofen itself is not part of our compound now, we cannot really tell that, oh, we have gotten better compounds that with better features, right? So
Okay.
yeah, yeah. I really appreciate your effort. You have really done well. Congratulations once more. I'm happy seeing all this.
Thank you, sir.
So you can stop sharing so that Dr. Joseph O can take over the stage for his presentation. Thank you very much.
Okay, sir. Thank you, sir.
So, Dr. Joseph, you can go ahead. Hello, Doc, are you with us?
Hello,
Oh, yes, sir. You can share your screen.
I'm trying to do that.
Oh, okay, sir.
I
Okay. Could there be any challenge so we can assist? Yeah, we can assist if yes. Yeah, I can hear you.
Hello.
Is there a challenge so we can assist you?
Hello.
Yeah, we can hear you.
Hello.
Yeah, I can hear you, but we are not seeing your screen.
We
Like
can hear you.
Network.
we can hear you, but you have not shared your screen. So if there is a challenge, you can assist
Hello.
me.
Hello.
Uh, yeah,
Hello.
we can hear you.
I'm trying to share, but I'm having issue with the sharing.
Okay, okay. Can you share the issue with us so we can see how we can assist you? What is the issue?
I want to suggest whether you can help me share at your end so that I just explain.
Okay, okay. I will do that. Why I'm doing that? So let the next presenter come in while I download your slide. All right.
All right. Thank you.
All right. Thank you. So, Mr. Salusi,
Yeah,
Mr.
yes, sir.
Yeah, you can now share your screen.
Okay.
Thank you. Okay. Okay. You can keep your phone at landscape view, so you can rotate it. Good. Perfect. Perfect.
Yeah. Good evening, sir.
Yeah. Good evening.
Thank you so much, sir. I just use this title as the work, my work as a technical screening and admin profile of as a mental therapy. Uh, as you people who as you can see, it's also one of the major neglected tropical diseases which affect thousands or even millions of people, especially in Africa. And the world standard to deal with issues is transcontinental. And the target is usually UKR project is Venus Guinness Research. So in my experimental workflow, number one, I did a short term where I identified over 2,201 transcontinental derivatives. After that, I utilize open Babel for analyzing the molecular representation that is the signal for course. After that, I then utilize Admit Lab 3.0 in order to predict the molecular modeling features. The features here I selected Lipinski rules of life, air safety, toxicity, carcinogenicity, neurotoxicity, ototoxicity, hematotoxicity, nephrotoxicity, and genotoxicity. So these are the features I selected as my benchmark. Here I utilize Excel, which we observed during our class, where I use each formula. That is, but in this case, what I 2001 derivatives, when I chose 0.5 as a benchmark, none of these derivatives comply with my benchmark. So I have to increase it to 0.6. Again, none of the derivatives pass. I say, okay, let me use 0.5. So after I use 0.7, like the pieces of out of 2201 pass my criteria, which I which I don't know if I was on right track. As you can see from the chat, my initial library contains about 2201 derivatives. Only 56 were able to make it. So it means here I have 2.5 processor range from my initial library. So imagine if I were to utilize with lab, billions of dollars would have been spent. So this is one of the reasons why we should adopt SQL tools to perform thousands of or billions of dollars. So up to 2001, only with experts and the success rate is just around 2.5%. Which if you were in Westland, there should be a loss. I don't think there is any serious company or big pharmaceutical company that can support such. So we are going to utilize 2001 derivatives, and out of them, it's only 50 pieces that will make it, which is the light of the day. Which is different if it's just 2.5. So that is it. So in my acquisitions, that I have already sent you, I have like a full Excel sheet, I mean, full approach sheet, in which there is my course of the previous sales related to compiler successful candidates, which I should move. So in conclusion, the integrated computation approach successfully reduces a large library of 201 transcontinental derivatives, in which only the pieces were able to see the light of the day, which shows us the optimal drug-like properties. Then the selected analogs demonstrate excellent predicted safety profiles. If that is the condition, is if my benchmark of 0.7 is accepted. And as you can see, I utilize Animate Lab 3.0, in which it is the one that I utilize to select my existing features. So my recommendation is this. Now the recommendation is this, that I should now take this 56 successful analog to study the binding affinity by performing molecular dosing. After that, I should now attempt to perform my study for performing candidates with this dosing score. After that, then we now move to in vitro validation. So these are my recommendations. Thank you so much.
Okay.
Hello?
That is a wonderful, yeah. That was a wonderful presentation, and it was impressive. I don't know. Yeah, does participant have question for him? Okay. Salisu, you have done a good job. But it seems you also reduced. How many compounds did you got? How many analogs were there in the pumpkin?
So they are 201 only.
Okay, but you reduced to 2,000 also.
Yeah, I use the number of users because they are less. So I just use them the way they are.
Okay. Oh, that is good. I'm really impressed. So at the end, you got 56 compound that uh, actually fits your model, and that really implies that if this 56 compound should now be taken into docking, we should expect a better result. So just like I have commented, I gave that comment to Daniel. The fact remains that when we are doing training like this, we may not really unleash everything. But we should, if we are doing screening, then we should also have a control. The control will not be the heat ligand which you developed analogs from. So that heat ligand will not be controlled. So we will now estimate to see if these ADMET properties of the analogs, if they were better than the ADMET properties of your heat ligand. So that will make the work to make more sense. Yeah, and that will tell us that the analogs are far better than our heat compound, even when we have not done docking. Yeah, this thing we are doing now, I used it this week. Somebody brought a work to my lab from Chukwemeka Odumegwu Ojukwu University, Anambra State. That work was on stroke. Stroke. Is a master student on stroke from biochemistry department. So she's working on 15 receptors. You know, when you are doing docking on 15 receptors, and then she has 5 medicinal plants, she was able to generate 500 compounds from each of these medicinal plants. Just look at how voluminous this work will be. So I was one that designed the work for her. Yeah, I designed the work for her. And if I'm designing a work for you, just be rest assured that you are not going to sleep, because I will design it in a way that even your supervisor will have mercy on you. Yes, so imagine those work that even your supervisor will be beating you. So I told her you will get 1,000 compounds for each of those your plants. So she has 5 plants, but she was able to get 500 and more for the 5 plants. So she's working on these 5 plants against 15 receptor. So I told her, we are not going to work with this 500 now. So we screen this 500 using what we did, this ADMET screening now. So we screen each of those 500. At the end, we got about 20 that passed our own model. When we did that docking, we finished some docking today before I came back home for this defense. The results were so marvelous, because at that point, there was no plant that gave a binding affinity lower than -8. It was from -9, 10, 12 above, because we have already filtered the ones that does not have the drug-like properties we need in that study. So this will limit you in wasting your time screening all the 1,000 compounds. So when I tell students that we need 1,500 compounds, they will think that we want to dock this 1,500 compounds. But this ideology now will help you to screen your compounds. So thank you very much for that wonderful presentation. As time goes on, we will keep learning. So at this point, I will.
Yes. Yeah, so I don't know if my benchmark I use 0.7 is also accepted, because during our training you only use 0.5. Even Mr. Daniel that has presented before me, he used 0.5.
Okay, you will answer that question now by yourself. You use 0.7, 70% failure. 70%, yes, 0.7 means 70% failure. So you you assessed students based on 70% failure instead of 0.3 means 30% failure, while 70% success. So you assess them with 0.7, that is 70% failure with 30% success. Is that a good way of assessing students to get brilliant
No,
students?
no, sir.
So if you want to get brilliant students, you subject them at 70% success and 30% failure, or 80% success. Me, if I was so wicked, I would keep them at 90%. So those who cross 90%, I will pick you. But unfortunately, those kind of people are not found on earth. Sometimes you have to mold them by yourself. It's just like a lady looking for a perfect man, which is not found on earth. She have to use sand and water and mold such person by herself. So I would suggest, we are told that 0.1 to 0.3 is safe. 0.31 to 0.5 is moderate. Then 0.51 to 1 is risky. So you can choose moderate or very safe. Yes, you are a good teacher, because when a good teacher sees that most students are failing, do you know what she will do? Mostly all these women teachers, feminine teachers, they have mother hearts. They will increase the success rate so that much students will pass. That was what you did. No wonder you got 56. Assuming you use 0.3, you would have found only 10 success. But
Sir,
nevertheless, you know what you are doing.
I use 0.5, none of them.
Congratulations. So
Thank you, sir.
you know what you are doing.
Thank you very
All
much.
right. Dr. Joseph, I'll quickly share your screen now.
Thank you.
All right. Can you see your screen?
Yes, I can see.
All right, sir.
Good evening, everyone.
So you'll be telling me, I'll be working with you. Tell me next, I go next. All right.
Thank you very much. So I
Yeah.
titled this mini project as Predetermined Criteria-Based Screening of Butylated Hydroxytoluene Derivatives in Silico. Next. Introduction. BHT is butylated hydroxytoluene. Another name for it is dibutylhydroxytoluene or butylhydroxytoluene. The IUPAC name is 2,6-dietert-butyl-4-methylphenol. The compound CID 31404. The molecular formula C15H24O, while the molecular weight is 220.35 gram per mole. This chosen compound is a lipophilic organic compound, and it can be synthesized in the lab. It is tasteless. Next slide. Uses of BHT. This compound is used as an antioxidant in food industry. It can also be used as flavor enhancer. In cosmetic industries, it is used as preservative. It can also be used in the treatment of genital cold sore that is caused by a species of virus called herpes virus. And this compound can also be used in the treatment of acquired immunodeficiency syndrome, which is AIDS. Next slide. This is the structure of BHT. The first one is the BHT in 2D. The second one, that BH, it should be BHT, not BHT. BHT in 3D. Next. The workflow of the project. Step 1 has to do with the choosing of the concerned drug, which is BHT. Step 2, derivatives were downloaded from PubChem, and I was able to see 8,802 derivatives. The SMICE were generated and eventually converted through Open Babel. Features gathering were carried out using AdmetLab 3.0. So that was done for
Sorry, can you still see my slide?
I'm seeing it now. That was done for the whole compound. Then the selection of molecular features were carried out. So at the end of the experiment, sorting of the successful compounds was done. Next slide. So these are the predetermined criteria that I made use of, and they were 8 in number. Number 1, I considered Lipinski's rule of 5. I considered F50, which is oral bioavailability. I also included blood-brain barrier. So I wanted to know whether those derivatives will be blood-brain permeant or not. So I also looked at carcinogenicity, neurotoxicity, hematotoxicity, nephrotoxicity, and genotoxicity. Next slide. So the total from this result now, the parameters and the figures. So the total number of derivatives were 8,802. So after getting that derivatives as our instructor taught us, they were in 2D. So I had to pick, no, he said some of them will be in 2D, while some will be in 3D. So I changed that portion to 3D conformer. Having changed it to 3D conformer, I was able to get 6,855 compounds. So the total number of the compounds that pass the general admit now, that means that was when I was screening them using AdmetLab 3.0. Not all of them came out. So only 6,843 came out, while others did not come out of the 6,855. So at the end of the final screening, only 115 passed the criteria set. Slide 8. Next slide. Conclusion. This study shows the 8,802 derivatives or BHT, out of which only 115 passed the set criteria. Next slide. Next slide, please. Recommendations. So I hereby recommend the following: that actually the full admit was done, but not really reported here. So number 2, molecular docking can be carried out. QSAS can be carried out, as well as MD. And why are we doing all that? We are doing all that to make sure that we are able to identify the least compounds for the purpose of drug synthesis. Next slide. Thank you for your attention.
Wow, that was a very wonderful one. I used to hear about BHT, but I never knew. What really attracted you, Doc? What really attracted you to BHT?
Actually, I worked on some plants, medicinal plants, sometimes ago, and I was able to, I did the GCMS analysis, and I equally did the HPLC analysis. So from the GCM analysis, I discovered that BHT happened to be one of those compounds. So while I was just scrolling down those past results of my GCMS, I just fell in love with this BHT. I know fully well that it is an antioxidant, so I just felt I should use it for this mini project.
Yeah, yeah. Because we usually use BHT as one of the control in antioxidant evaluation. So it simply means that if this your 115 compounds, we are better than BHT in terms of ADMET properties. That means we may be on the pathway of getting better antioxidant drug, because what we recommend mostly for people to take is drugs rich in antioxidants. That is why if I want to develop a herbal formulation for you, I must include a plant that is rich in antioxidants and also a plant rich in anti-inflammatory. These two components must be found in your herbal formulation. Even if there is no anti-inflammatory, but antioxidant must be there. And because most of our disease arise due to free oxygen radicals, ROS. So antioxidant is very, 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 very important as far as life is important. We encourage you to eat foods that are rich in antioxidants, fruits, vitamins. Very, very good. So I really love this choice of antioxidant. Thank you so much, Doc.
So
Thank
this
you.
work, I don't want you to stop here. I want us to do something reasonable about it. Just as you were mentioning herbal formulation, we should do
Yes.
something about. I still have the other compounds that I just pick only one out of that work. I have up to, if not up to 100 that were analyzed. So I don't
Yeah.
know how we can do something and bring something reasonable out of it.
Okay, we shall discuss behind the scene so we know how we can come into that work.
All right.
Thank you once
Thank you
again.
very much.
Yeah. Uh, I will stop sharing my screen so that the next presenter, Akintoku Emmanuel, he took permission. Okay, Abiodi, your hand is up. Sorry, I didn't know.
Yes, sir. Good evening, Dr. Harry.
Yeah, good evening.
Yeah, based on the presenter, Doctor, I
Yes,
love the presentation because I've been working on antioxidants, anti-inflammatory, and basically cancer, cervical cancer precisely. And I've been
okay.
using BHT as a standard and used to have a very low IC50.
Mm,
So, and
mm.
based on that, that means the lower the IC50, the more
Yeah,
potent the compound will be.
yes,
So basically, like 10. So it used to be the best. So this would be a very good job if you can see a kind of derivatives of BHT. You know, we also use vitamin C and the likes as
yes,
standard, but
yes,
BHT is very common as the potent compound with antioxidant properties and
yeah.
as a reference, commonly as a reference. So it will be a very good job to develop this particular project to bring out the best from this, sir.
Very, very good. Yeah,
Thank you, sir.
very good, very good. Okay. Uh, yeah, Dr. Abisoye, can see your hand up.
Good evening, sir.
Yeah, good evening, Doc.
Yeah, thank you for the presentation. So I want to ask that, is there a chance that some of those 2D compounds that were excluded because they were not 3D compounds would maybe might have given better results than BHT?
Very good, very good, very good. Yeah, there is a chance. There is a chance. That is one of the limitations we have in PopCam, because when immediately you download those structures, you are not the one that remove the 2D, because PopCam gives you option to choose 2D or 3D. I must choose one. So when you choose 2D before downloading, it gives you 2D. when you choose 3D, it gives you 3D. So I think that to boycott that, after you download the 3D, you also choose 2D and download the 2D to complete the number that was written there before you download. Because 2D compounds, they are not impotent. 2D compounds are not impotent though. It's just that they are in the 2D format, which we have done. I exposed my participants in the 1 month training on macromocy. I had to show them how to convert 2D structures to 3D. So even if you achieve those 2D structure, you can now convert those 2D to 3D. I showed them how to do that. So, Dr. Abisoye, your point is very clear, because those 2D structures we neglected might even be better. Yes. So what you will do is, when you take 3D, because the number will reduce, then come back again to that same scaffold and choose the 2D. Yeah. Okay. Dr. Ajola.
Yes, I have a question too.
Yes.
Yeah, please, sir. The network was a bit off, but one of the participants made a comment about, is it vitamin A or vitamin C being an antioxidant? Vitamin C, okay. But I didn't really get the point he was trying to make concerning the presentation and vitamin C.
Okay. The presenter used one of the reference I'm having echo. Just kindly mute your mic. All right. So the presenter used, that was the last presenter. He used one of the reference drugs he discovered in his plants first. That is BHT. That BHT was found in one of his plants. I'm coming. Just give me a little. Okay, so Dr. Joseph noticed that from his previous studies, one of the plants has that BHT, so he had to use the BHT to get the derivatives. Unfortunately, the BHT also is a reference, is a known antioxidant drug, so he had to find out the derivatives. So on the point of discussion now, Dr. Abiodun noted that in his own research that he has been doing on antioxidants, he has been using BHT and ascorbic acid as a reference drug for antioxidant assays, and that BHT has been far better than ascorbic acid based on their IC50 values. So he now encouraged that further work should really be done on that BHT. So that was where ascorbic acid came in, because ascorbic acid is also a reference drug for antioxidant. Is a big antioxidant.
I understand now. Thank you. I understand. Please, are we getting this recording as well, please?
Yes, and you're not paying for it.
Thanks
this one will be
much.
this one will be everybody. This one will be to everybody. To the platform.
Thank you, sir.
Yeah, thank you. So please, this your bad guys, people will not kill me. Akintoku Emmanuel took permission, unavailably absent. Professor Olabi Otimi, I don't know if he's here. Professor Wallaby. Okay. No. He's not here. Okay. Then the next is, please forgive me. The next is Beneme Azota Ogechi. So please, Mrs. Azota, you can go ahead.
This is Mr. Hey, well, hello. I'm here.
Yeah,
Okay,
go
so
ahead.
can I share my screen?
Yeah, share your screen and go ahead.
Okay. 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 So good evening, everybody. My name is Mbwemen Azuko Gete, and my topic today is titled High Throughput Screening of Curcumin Analog Compounds, which is a natural product, similar structure, via selected pharmacokinetic features, not spelling. Okay, so intro. So the curcumin is a phytochemical compound derived from the Curcuma longa, known as the turmeric plant, which is an Indian rhizomatous herbal plant of the ginger family, quite similar to ginger, based on how they are grown at the rhizome where the turmeric itself forms under the soil. So the medicinal benefits of turmeric is due to the presence of these active compounds, which are collectively known as curcuminoids, which include curcumin, dimethoxycurcumin, and this dimethoxycurcumin. And it's basically a yellow, the turmeric itself is a very yellowish plant due to the presence of carotenoid compounds in it, and it gives it the very obvious, pungent, strong yellowish color. So the turmeric or the curcumin is basically a known herb, a medicinal herb, which has majorly antioxidant, anti-inflammatory, anti-mutagenic, antimicrobial, and so many other anti-properties. It could also aid with, illnesses like arthritis and the likes of it as well. So it is, as I said, it's stable at the acidic pH of the stomach, meaning that if it could be consumed as a drug, it won't just disintegrate completely without activating its therapeutic effect once it is ingested into the body. And my workflow included first of selecting the desired compound, which is the curcumin, from the PubChem database via the selective or similar structure search. Then I had to download the whole 3D file, which sifted out the 2D file as Mr. Henry said earlier, and converted this FDF format into the SMILE format using the Open Babel software. After this, I had to generate the pharmacokinetic features of these converted SMILE compounds on the ADMET 3.0 database, and then selecting my specific features for screening and using it to pick out the total screened successful compounds at the end of the day. From the okay, I'll still zoom in, but before I zoom in, I'll say that the total number of compounds I screened were 4,441, and I used as the scales. I used the 0.5 less than 0.5 maximum scale and a zero point 0.4 maximum scale to deduce my final compounds. So out of the 4,441, for the 0.5 scale, there were 92, which were successful, as seen here. So the features included, before I skip to the success rate, included the Lipsinki's rule, which indicated no violations at all. Then the F50 indicating oral bioavailability for easy ingestion through the mouth. Then there is carcinogenicity if the compounds are, if they don't cause any form of carcinogenicity to the humans who will consume them. Then neurotoxicity having to do with toxicity against the nervous system and the peripheral nervous system. Then ototoxicity, toxicity to the inner membranes of the ear, hematotoxicity against blood, toxicity of the blood and bone marrows, nephrotoxicity, that is toxicity against kidney functions, and then genotoxicity, which is regarding to genes causing mutagens and mutations at the end of the day. So at the screening at zero point 0.4, there were a total of 19 successful compounds from the 4,441, which is a 0.5% success rate. Then for the zero point 0.5 scale, it was a success. There were 92 compounds which passed the zero point 0.5 scale screening, which is a 2.1% success rate. And from this other, I use this is my this particular singled out compound is the original curcumin smile code structure. And from the results, you can see that it failed the test, the original, but 92 of the total derivatives passed the zero point 0.5 and 19 passed the zero point 0.3. I also even did a did a zero point 0.4, and I also did a zero point 0.3 scale, whereby only 4 passed the zero point 0.3 scale, but I didn't include it here. But I just left that one to the corner. 4 out of the 4,000 passed, while 19 for 0.4 and 92 for 0.5. So these were the results of the screening features. And in conclusion, the high throughput screening just basically is a reliable method of scanning through large libraries of data to pinpoint selected or specific activities of this compound, because the whole essence of doing this is to check which of these large compounds have some useful biological activity and screening potential to be possibly converted into a drug if it is not a drug already. Then the features, we are to ensure there is minimal toxicity at the cellular, genetic, and also tissue level, and also ensure proper lipophilicity and absorption criteria for the derivatives, meaning that we could pass further screening tests for possible drug development, delivery, and even clinical trial experiments and testing. And I recommend that these natural products could be further assessed, their real-time safety profile and formulation capacity into actual drugs with specific target proteins of the host, because you can't just formulate a drug without having a target that you're trying to reach, and making sure that you're able to reach that target through safe drug delivery methods which will not harm the host you're testing these drugs on. Then there is also the techniques involved in formulation, in vitro and in vivo study analysis, and even docking and molecular dynamics simulations to tell how well these compounds are, ingested, how they are able to induce any therapeutic effects and make the host feel better at the end of the day. And as Mr. Henry has been hammering, we are always neglecting our herbal products. We are only based on foreign drugs and compounds, which are okay, but at the end of the day, if you break down these drugs, you find out that many of them are actually quite toxic, and they're just bunch of chemical formulations. And even if they are, some of them could be some biological formulations, but the chemical formulations could be more than the biological in terms of, semi-synthetic formulations of drugs. So yeah, I believe in herbal medicine, although I haven't tried it, but I believe in it. Yeah, I'm a scientist, so I understand that. Thank you.
Okay. I think I'm happy. Uh yeah, because at the point I saw where you used your curcumin, you
Yeah,
also screened your curcumin, and if I was not daft, I heard where you say it failed.
it failed.
So
Yes, it failed woefully.
now it simply means that from your screening, you're able to get derivatives that will be better than
Mm,
curcumin. Yeah, derivative that will be better than curcumin, and that is the whole essence. That is the whole
yes.
essence. So that is a good job. I don't know if participants have questions for him.
So this was actually quite stressful because I first started working with two drugs. One was an anti-malarial drug, then the other one was an anti-tubercular drug, but they were all, negative after screening over 10,000 compounds. I almost got blind. I exited and had to choose something else. I
Chai.
thought you were joking when you said the first time that we could screen over tens of thousands and everything would be negative. I thought it was a joke at first until I actually
Eh,
did it and somehow.
Thank God you have seen it.
Seeing is believing.
Yeah, Dr. Abiodun.
Yes, sir. I just want to commend the last presenter for a job well done because he was able to do a proper screening by comparing point 4, point 5, the
Yeah,
essence of the best four compound. It is a
yeah,
job well done. Thank you very much.
yeah.
Thank you. Thank you,
Our,
sir.
our only challenge in a synthetic screening like this is towards the end, because our end is now to
Synthesize.
synthesize. So that is
Yes.
just, but,
That's the main
but we,
challenge.
yeah, but we keep hoping. We keep hoping
Exactly. Exactly.
that
Exactly.
God will help us. There will be a way for us. I
Amen.
swear. Once I see a way of synthesis cheap and effective, that is when we start to enjoy what we are doing here.
Yes, sir,
Yes.
because it's real life practice, no more just sitting behind a computer.
You'll be seeing the
Uh,
best result that the synthesizer will not be the main challenge.
It's a problem.
yes.
Why is it always like that? These things are much easier abroad in different countries, especially Europe and places like that. For Africa, a different story.
Is well. Okay. The next presenter should be Dr. Oyelese. Sorry, Dr. Johnson. Better. So I will not spoil the remaining part of the name. Okay.
Good evening, everybody.
Yeah, please pronounce this Yoruba name. Olari
Oyelese, Oyelese,
Oyelese.
Oyelese Olanrewaju.
Hey, okay.
Thank you for this opportunity, Dr. Henry.
Yeah, thank you, sir.
Actually, I'm not yet a doctor.
You are
I'm coming behind all you people, believing
no.
that one day I will get there. But I've been enjoying this training all this while. And that's why I'm trying my best to be able to understand all you have been teaching us.
Thank you.
In the first place, I wasn't able to join through my laptop. I'm
Okay.
joining through the Android. But I
Okay.
wouldn't know if you'll be able to help me share the screen, if I'll be able to see, because all that I've been presenting all this while, network did not allow me to see what they have been sharing. I was just listening to them. But
Okay.
be that as it may, I will still explain the little I did.
Okay. So that means okay. Let me look for your slide,
Okay, sir.
so that it will be very easier for us once you are present. But will you be able to see the screen?
If I don't see it, I will be able to see it on my laptop. It's only that I could not join the something with the laptop.
But you are joining with your phone, right?
I'm joining with my phone. Yes.
That means your phone can see our slide.
Yes, sir. I hope so.
Right. All right. So I'm there already.
All right, sir.
Okay. Coming. Just. All right.
Good evening. No, I'm just joining the party. I hope I still have something to offer.
Oh, yes, Prof. Maybe after
I'm
now.
very sorry. I'm coming late. I really forgot.
There's no problem. But after
Thank
this
you.
presentation. Okay. Dr. Johnson, can you see my slide? You can go ahead. Can anyone see the slide?
Yes, you can see.
Oh, so Mr. Orile say you can go ahead.
I can't see it, but let me just be presenting for you and I will be asking you to change it for people to see.
Okay. All right. Better.
I titled it Molecular Modeling and Screening of Analogs of Fluoxetine. I came across the fluoxetine when I just browsed for some common drugs. I didn't have any compound of interest in from any natural source that I can use, but I choose that drug from a sample of compounds I browsed for. That was the one I used to get some derivative of it. Then I titled it Molecular Modeling and Screening of Analogs of Fluoxetine. Next slide, sir.
All right. Yeah.
Okay, sir. The introduction. Molecular modeling is the use of theoretical and computer-based mathematical method to simulate, visualize, and analyze the structure, behavior, and properties of atoms and molecules. I wouldn't want to waste much time on that because our instructor have explained all that to us in our trainings. The method are used in the field of computational chemistry, drug design, computational biology, and material science to study molecular systems ranging from small chemical systems to large biological molecules and material assemblies, and so on. Next slide to see the workflow.
Yeah.
Experimental workflow. Selection of synthetic molecule, generating the derivatives or analogs of the selected molecule, generating the SMI codes of those derivatives, getting the features of the compound using ADMET 3.0, selecting molecular modeling feature and benchmark for screening of the compounds, then screening of the compounds and sorting of those that pass the criteria. That was my workflow. Selection of synthetic compound. As I explained the other time, I just browse for some common drugs, out of which I choose the one that is of my interest and the one that have reasonable number of derivative. I choose fluoxetine. Fluoxetine brand name is Prozac. It's possible some of us might have come across it. It's a type of antidepressant that works by increasing serotonin levels in the brain to improve emotions. It's a drug that is used to improve emotions when someone is depressed. Uh, I didn't include the molecular weight, the CID, and molecular formula. It was when I was listening to presentation of Dr. Joseph that I knew I supposed to add them to the presentation. Pardon me for that.
Yeah, no problem.
Okay. Uh, I choose that and I went to PubChem to generate the derivatives. I have the derivative separately that are in a folder that I sent to you. I was able to get 2,304 derivatives, out of which only 2,040 were 3D molecules, and those were the one downloaded. Because of the problem of power supply here, I wasn't able to screen everything. It was even this evening that I finished the screening because whenever I exhausted power on my system, to continue the work becomes difficult. But I was able to screen 503 out of that 2,040. I generated them. I generated the SMI codes, and I subjected the compound to ADMET Lab 3.3, 3.0 screening, in which I screen. I choose less than 3.50 as my benchmark. I screen them by Lipinski rule, the bioavailability F50, carcinogenicity, neurotoxicity, and all those ones that the instructor used for us, about 7 of them. Uh, Having screened them, I used the benchmark of less than 0.5 to screen all of them. But I discovered that all of them failed the screening. In fact, I was disturbed until when I was listening to Mr. Ngbeneme that said that he has tried some like that, that all the 4,000 failed. But in my own view, what I felt when all of them failed was what I saw in my something, in my conclusion, that I concluded that all the analogs that I generated, that I downloaded from the PubChem, they are not potential drugs better than fluoxetine that has been produced before. I wouldn't know, maybe if I've been able to screen all the 2040, maybe I will have obtained one that will be positive. But be that as it may, what I recommended, or let me say to myself, or I thought, had it been I have enough time, was that I would have tried to generate my own derivatives of the compound instead of downloading from the PubChem, just like the way you taught us. Is it not on? Was it not under pharmacophore design that we can
Yeah.
design, we can derive our own derivatives? Then that was what I thought I would do later, that I would try to generate derivative myself and now screen them and see if I will be able to get any of them that will pass the screening. But for all the 500 that I screened, all of them failed, which means none of them is a better potential drugs than the fluoxetine thing. That was all I did. Thank you, sir.
Well, that was failure is a result, and it's failure is a good result. But people don't like to have failures as a result. But it's a very good result, and that result means that it's where it's where it's good when you encounter stuff like this, because you notice that these were analogs generated and just uploaded. Most times, these analogs they were not modeled based on any condition. They just upload them. So we now say, okay, you can model this by yourself. You can model this by yourself, and thank God for what is happening to. If the mic is on, just let me mute you.
Okay, sir.
Yeah, so thank God for what is really happening to those who joined us last week of January in the beginner's course. They are building 6,000 analogs manually. They are using ChemDraw to build. I believe before they will be true, they will lose body weight. It's just like when you are assessing effect of a liver extract on the body weight of albino rat, and you notice that there is a reduction in the body weight due to stress. So similar thing will happen to most of them. There will be reduction in body weight. Imagine when one person is creating, not downloading, you are building for yourself 6,000 compounds.
Sometimes I'm also included.
Maybe
I'm
I
also
will
involved.
increase that job. I
I
will
don't
increase
increase
the
it
job.
now.
Good. I will increase the job. Something just crossed my mind, because their job was just to.
If you increase it, is December will present.
No, their job was just to model.
Don't sit it in December.
What if after you model now, you do screening? You do screening with them. It will be better now.
I'm going to do
You
this
do
screening.
you do screening with them.
Exactly.
It just came, a thing just entered my mind. So you don't just end the modeling. You just try and do screening with them, so that you know how well you have modeled this compound.
All right, sir.
Yeah, the screening is not a small job. It's not a big job again. We don't pass that level. Okay, thank you so much for that presentation. It's a wonderful one. While we are on, Dr. Utimi join us. Sir, I don't know if you are still here with us.
Yes, I am.
Okay. So will you be, I don't know if you are ready to share your screen with us.
Okay, I'll do that straight away.
Okay, sir. Thank you.
Yeah, can you see my screen?
Yeah, it's coming. Good. Wonderful slide.
Yeah, so I'm presenting. May I start?
Yes, go ahead, sir. You have
Yeah, I'm presenting the computational screen of aspirin derivatives for some drug absorption and toxicity and safety parameters. My name is Owolabiru Timi from Obafemi Awolowo University in Ife. Aspirin, popularly also called acetylsalicylic acid, is a non-steroidal anti-inflammatory drug that is one of the frontline drugs to relieve pain, fever, and inflammatory conditions. Because of its ability to inhibit platelet aggregation, it is also used as a blood thinner in conditions that are associated with high risk of intravascular coagulation. The mechanism of acetylsalicylic acid is linked to the disruption of production of prostaglandins throughout the body. It is known to inhibit the COX-1 and COX-2 enzymes to produce the biological effects that are highlighted above. Okay. The absorption of aspirin occurs mainly in the stomach and the proximal section of the GIT. The absorption is associated with gastric irritation, with possible ulceration, and this has been attributed to a phenomenon called intracellular ion trapping. The drug is generally considered to be a safe drug. Because of that, it is available as an over-the-counter drug. And this has made it easily accessible to people, and this easy access has increased the risk of misuse of the drug in the general population. And there have been reports of accidental and deliberate ingestion of overdose of aspirin, and this has been associated with serious consequences like liver failure and neurotoxicity. Also, aspirin is often used by people with chronic inflammatory conditions for a prolonged time. And the prolonging of aspirin has also been reported to increase the risk of analgesic nephropathy. So this study set out to screen the derivatives of aspirin for their suitability as drug candidates on the ADMET class screening platform using some oral absorption parameters. We also screened for a number of toxicity parameters in these derivatives. So what we did was that we downloaded the aspirin derivatives on the PubChem database using the similar structure search menu. The list of the downloaded derivatives were saved in their structured data format. The downloaded data was imported into the Open Babel software to generate their SMILES code. And the SMILES codes were later transferred to the ADMET lab to generate their physical, pharmacokinetic, and toxicity features. The derivative was screened for the parameters of interest, and in this study, the parameters that we screened for include that is the absorption profile. We use the human intestinal absorption, the F50, which is the 50% of the bioavailability, and the drug-induced liver injury toxicity. We also assess for neurotoxicity and nephrotoxicity using a cutoff of 0.3, and we did this on the Excel workbook. The results, the PubChem results yielded 6,903 derivatives, out of which 78, which represents 1.13% of the derivatives that passed the screening criteria. So in conclusion, we concluded that out of the 1,609 aspirin derivatives that we derived from PubChem, only 1.13% of them, which is equal to 78 derivatives, have favorable absorption and safety profiles. We recommended that the derivatives that pass the screening test should be subjected to further screening for drug likeness, docking studies, and in vitro and in vivo validation of these computational results towards proposing them for drug development. These are my references. Thank you.
That was a very precise one, and all the process we are carefully taking. The result really shows that there could be compound that may be better than aspirin in safety and in efficacy. Because when you look at the combination of the criteria, it combines both binding features like the human absorption, so absorption features and the toxic features. And I appreciate seeing 78 compounds out of 6,000 plus compounds. So this process is really, really impactful before you proceed to your docking. Like I always say that when I look at docking papers, you hardly see people who do this kind of pre-docking screening, because everybody just take what they have in docking, go to docking, because docking has become common. So docking has become so common that everybody can do docking. But the main issue is now your workflow, the protocol you developed for yourself in your work. So I appreciate all the presentations. They have been good. They have been good. Like as we said, just lastly, that we pray that God will answer our prayer to give us a place where we can do these synthesis at will without spending a lot of money. We are still looking forward, HENDEP is still looking a way of collaborating with private institutions outside the country that can do this synthesis very well, because I have seen a lot of work on synthesis. When we had the training on chemoinformatics, the literatures we are using to build our own database were job done through synthesis. Those literatures were people who did synthesis, went to the lab. So these people modeled the compound. If you look at those literatures, they modeled the compound, got about 5 with their label, probably 13A, 13B, 13C, up to 13F, according to their label. Then took those pure compounds that they will now went to the lab to synthesize what they modeled, put it to the lab and did the in vitro study. That is a full work. So I'm still looking for a way of contacting some of those journals, the authors, see how did they really do this, their synthesis. Let's see, because we need to also do our own synthesis. We need to start synthesizing. So that it will not end in theory and in silico. So definitely, I'm going to look out for a way out. Yeah, the content I had in Brazil is too expensive. And even one of the work we sent to Brazil for synthesis, the product given to us, if I tell you that we have it again, no more, because by the time we begin to take some portions of those products to do in vitro work, we exhausted all the products. Because it will become difficult for you to start doing synthesis on a high percentage yield, giving high percentage yield, high amount. To achieve high amount of your product, high quantity of the product is another challenge you have. But I must definitely look out for a way where we can see reliable synthesis. Once again, we appreciate all your efforts in being around.
Sorry, Dr. Henry, talking about synthesis.
Yeah.
Well, I am not a chemist, but I remember a couple of years ago that I co-supervised a work in my university, and we used a pure compound that was synthesized. In fact, two works. One was as an antidepressant, and the second one I can't remember very well. I'm not in Nigeria now, but I think I can still get in touch with those guys that did that work. The one for the antidepressant was synthesized, I supervised with the guy that you were mentioning yesterday that submitted a protein to the PDB database.
Wow.
Yeah. Yeah. He was the one I co-supervised a work with him on antidepressant. In fact, they synthesized a derivative of fluoxetine.
Wow.
You know, but I'm not, the only problem now is don't know whether that synthesis was done in Nigeria or South Africa. I will confirm and let you know.
Very good. Very good.
I will confirm. Just keep me reminded. That's one. Then two, I will also ask somebody that I know works in a university in the UK. The person is not a chemist, but I will ask if the person can help me ask their chemistry department or faculty, or if they have a facility to synthesize in that university. The person is a staff of the university, and I think we can leverage on that. I'm not a chemist. I don't do much of chemistry work, but if this kind of demand is coming up, I think I will do what is within my capacity to help if I can. So I will again probe the guy in my university in Ife and also talk to the person I know in the UK.
Thank you so much. Thank you for that selfless heart to help us out. See, I was even mentioning somebody, I don't know that. That is what they say, that the word is a small word. I was mentioning somebody that uploaded work, but I never knew that one of his co-author in a previous research work is already here with us. Because when I see good things, I love to say it out. And it was one of our participants here that also shared that post with me on LinkedIn. That is Mr. Salusi.
Yes, sir. God.
Yeah, he was the one that mentioned that, shared that to me, and I was marveled to see the protein. I've actually studied the protein, and it's good. It's good. So that is what we mean by products, because product now must not only mean having a physical or a tangible item, but his research work has been translated. You know, what we are preaching now, everywhere you go now, you hear translational research, and that is what we are preaching. That is the ball now. We are talking about translational research. So we don't just end in paper. We don't just end in paper. So once again, I appreciate everybody. I'll just take Christos. I'm seeing Joseph.
Yes, thank you very much for today. I pray the Lord will strengthen you the more. Please, I want to say some things, and I would like Dr. Rotimi Owolabi to please pay attention to what I want to say. I don't know whether he's still around with us.
Yeah, I'm here.
Okay, sir. I remember that sometimes ago, while Professor Agbedansi in Ife, that headed the drug production unit, was alive, I remember that some products came out from that department, herbal products, like something Mama, Jedi Mama, something decoction like that.
Jedi decoction.
So, Something like that. I remember about 3 products came out during that time, and those products were able to be NAFDAC at that time. So I want to ask now, how easy can it be if somebody has the mind of going into something like that to produce herbal product like that? How can you possibly be of help? That's my
Well,
question, sir.
okay. Well, yeah. I am not in DRPU. DRPU is the Drug Research and Production Unit of OAU, but I have colleagues that work there. I knew the late Professor Agbeda Onsi of blessed memory. May his soul rest in peace. So it's
Amen.
so it's a department university that I have very good relationship with. I will, if as I said, currently I'm not in Nigeria. I should be back to Nigeria in the next 1 month. So I can share my telephone number and my email address with you. You know, we have a lot of things in our head, so that I don't forget. Maybe around middle of March, if you don't mind, you can just, shoot me a mail or give me a flash or a call. Then I can help you find out, if they have such a facility. I'm not sure they have a synthetic facility, a facility to synthesize in DRPU. The much I know, I don't think they do, but if I know about the jadid decoction, and I know some other products that were developed, in the faculty of
Mama
pharmacy.
product.
Yeah. I know a lot, so I can ask, the people that are still there. I'm sure they have an idea of how they were able to get that done, and we can take it up from there. I hope I answer your question.
Yes, sir.
So, well, what's it called? I'll provide my email address on the platform. You can pick it up, or on our various WhatsApp platforms. I think I'm in almost all of those platforms. You can pick up my phone there as well. So as I said, maybe middle of the month of March, just shoot me a mail or give me a call. Then we take it up from there.
All right, sir. Thank you, sir.
Yeah, thank you. Yeah.
All right, Mr. Salusi.
Yeah, yes, sir. So I have a question that I willing to ask you, although it may be out of context. Into this question. So my question is this: I like recently that post, LinkedIn post that I had you read about the new implicated protein that was isolated and deposited in the protein data bank. So the question I have is, like for a malaria, we got to understand that we have more than one implicated protein that is believed to cause
Hello?
Yes, sir, can you hear me?
I can hear you now.
Yeah, I was saying that if you look at malaria, we already have more than one protein that are implicated to cause malarial infection. So does it mean
Yes.
if you discovered a novel small molecule that combine to a particular implicated protein? So what are the other implicated proteins? Does it mean that the same molecules you discovered can target all the implicated proteins?
Okay. Now, one, a molecule discovered cannot target all the proteins. The malaria you see has multiple proteins. Some are not yet discovered. Some are not yet discovered. So I believe what those guys did was, from their studies, from their genomic data, they were able to discover a set of gene that has not been reported. It's a set of gene that is associated with malaria and yet have not been folded into protein, has not been reported anywhere. So they have to develop that protein from those set of genes. Then they have to also look for a drug that can be used to target that protein much more better than other drugs. So that was the whole idea of just how the work revolves. But like I'm saying it now, it may look simple, but it's actually a big work because you have to do a lot of work on multi-omics. You have to involve multi-omics in that study. Multi-omics must be there, and we don't have those equipment in Nigeria. How many do we have Sanga in Nigeria? I don't think if there is one Sanga machine in Nigeria. If there is, I may not
I
know.
think
I think
is there no Sanga machine in IIT? I think there should be one in IIT.
There's one in Junior Osun as well.
Oh, they do. Wow. Then
Yeah.
why then do people always send their sample to Inkwaba?
I think
Maybe they don't have that
can I
information.
come?
Yes.
Yes. Thank you. My BSc project, I work on malaria as well, and I use a particular plant. And after the molecular docking, I did not do the MD simulation because of the level. So after the docking, I found out that there were 3 heat compounds that perform far better than attesulate and chloroquine.
Yeah.
But there's one particular thing that I gained during this program that I will never forget. It's very good to do toxicity before the molecular docking.
Of course.
Very, very good to do that. So, and part of my colleagues, some work on, I work on LDH, lactate dehydrogenase enzyme. So this is a particular enzyme used by these Plasmodium falciparum particularly. You know, their only means of survival is through glycolytic pathway. And the end product of the pathway is pyruvic acid, pyruvate. And being a condition that occur under anaerobic condition, one of the fate of pyruvate is that it will give us lactate. And the enzyme used there is lactate dehydrogenase enzyme. So whereby they will generate NAD+, they oxidize nicotinamide adenine dinucleotide that will help us to generate ATP continuously for their survival in the system, to continually consuming red blood cell. So then other colleagues work on other protein. We are about 3 or 4 at Summit University of Akwara State. So later we compared the results. We found out that all these extracts, they work separately and they work best based on their target and the reference drug used.
Yeah.
And let assume after the work, if we are able to proceed on the work, move further, we can even discover a particular protein that is novel.
Yeah.
If you work harder on it, then after that there will be a lot of molecular techniques. But presently I'm working on cancer for my MSc. Basically, I focus on molecular biology aspects at Frontier University of Osogbo. And the Frontier University, we have a lot of equipment in molecular biology. So probably I'm trying to make reference to Dr. Imran question.
Yeah.
So for us to we made mention of homology modeling. So that will be part of the method used for them to be able to work on that protein being deposited or PDB. So they are going to pass through a lot of molecular biology techniques. Majorly, they focus more on genomics. And they have to do sequencing because I attended the training in Osun, I think last year, December. We worked majorly on Sanger sequencing. They had Sanger sequencing. It was collaborated with Nkaba. The two of them work together, and they trained us very well on Sanger sequencing, how to design primer, and a lot of other molecular
tools. So
Yeah.
after the training, we discovered that after discover a particular gene, you can go with sequencing. Then after through with the sequencing, we can now go into this homology modeling. That's the under bioinformatics aspect.
Yeah.
So I think by at the end of that, we can do a lot of things just to proceed on that particular protein being discovered.
Wow, that is great. I just heard that Ocean has, is it I want to be clarified because I want to stop sending samples to South Africa.
Now they have it too.
Yes, so they
And
have
we're
a
even
Sandra
giving
mission.
yes, yes.
Okay.
And they are multipurpose research lab. They are
Wow.
MRL, multipurpose research lab. I think
Okay.
part of the team is Professor Masuru, the DVC of Federal University of Science, I think Ilarogun.
Okay.
That the
You are sure.
present part of their chairman presently is Professor Lukman in chemistry department, the University.
Okay.
I think is the head presently.
It seems that Ocean State generally, all the schools in Ocean State, they are really doing well.
They are trying. They are trying.
I'm beginning to get records that most of the schools there are doing well.
Very soon
I
we're going
really
to have that
appreciate
too in my school,
Yes.
Fountain University. Presently, we have a convocal microscope and we have a lot of PCR, qPCR, and the conventional PCR.
Oh, that is good. Okay. So that was
And
a very good
the
contribution.
Center of Excellence and the Redeem University too. I think they have a number of sequencing facilities there too.
Yeah. Because
Yes.
I know
Yeah,
that also
Professor
Happy is doing
Professor
so
Happy,
much.
yes.
Yeah, Happy is doing so much in my university. He's really doing
Yes.
so much. And my prayer is before this set of people, before they will leave, they should help transmit this knowledge to the younger generation. So because if you look at their age now, in the next 30 years, you may not be seeing them again fit in the academics. So I really love what Professor Happy is doing. He is part of the people fighting for this African Genome Project. And they have been doing well. They have been doing so much well. As I listen, I think your hand is still up, or the former hand I was up.
Yes, sir. So I wanted to say, for instance, if you go to hospital now, if you count 10 different people that have been diagnosed with malaria, you find out that they have been prescribed with the same anti-malarial drug. And
Yeah.
maybe once they take that drug, maybe more than 100% of them will be treated. So what I mean, the present anti-malarial drugs we have, are they multi-target? Because I can't understand that if I were to go to hospital, the same drugs will be prescribed me with one of my friend who is also malarially infected. Does that mean our malaria, the target, the implicated protein that found in my, let's say, in my present condition is similar or the something with other guys? That is my question, sir.
All right. I will come in quickly to that. There is a project ongoing also on personalized medicine. The project is ongoing on personalized medicine. And that was why I asked for Sandra machines, because abroad, they're already practicing that personalized medicine. I was discussing with a friend of mine just early this week that in some developed countries now, if you have malaria and maybe both of us have malaria, then we have to visit a hospital for malaria and complain that we have malaria. Now, before they will even give us that drug for malaria, they have to do a genomic study. They have to take samples from us, analyze our gene immediately, find out the variant genes that they will end up. It's possible that even both of us have malaria, but the genes that will be expressed there must be one or two that changed in our system, because our own genetic, our own gene, our own body has influence on all this disease. So that they may not give us, I and my friend will not be given the same drug. They have to look for a drug that is compatible to my own genetic information that they got, and they will give my friend another drug instantly. That is the whole idea of precision medicine or personalized medicine, where we have one person, one drug, one person, one drug. So there will be lots of drug, not that 1 million person will not have 1 million drug. So they might not look for the drugs available that will be compatible for that particular person. Now, I may agree that 100% might take Leonard and they will get healed of their malaria. Yes, but it's not effective. It definitely will not be effective. It will not be effective. Out of that 100 persons, the ones that might be effective might not even be up to 50, because there must be variations in the malaria targets. There must be variations. If 2 persons are having malaria, we will not have the same malaria target. There must be little variation. If you do the whole genome study, there must be little variation. So it is that variation that they are looking after in personalized medicine. So, but to you, you all take Leonard and will get healed. But there are cases whereby people have taken a drug. They keep taking a drug. It's as if that drug is not working for them. But that is the drug they prescribe, but it's not working. So all these are reasons why people are now coming up on personalized medicine. The project is a heavy one, is ongoing. One person, one drug. A drug that is suitable for your whole genome. So I agree, as I listen, that 100 persons might take that same drug and they will get healed, but the effectiveness, the safety, efficacies will not be the same in this 100 persons system. So because we don't have availability of standard machines, because you need a machine that you do instant work and you get, my dear, personalized medicine is the best. It's just that we are in Africa, but it's the best. Some parts in South Africa is already practicing personalized medicine. Just little part. They are practicing personalized medicine. Though it's expensive, we need equipment. But we'll get there. It is the best. It is the best.
It is.
So we really want to appreciate. I have another session by 8 o'clock, and this is already 10 minutes to 8. That means I will just do about relay race, where we immediately switch over to that other class. Once again, we are all welcome. I want to remind us that how many of you have seen our flyer for the webinar next week, Wednesday?
Yes, I have.
All right.
I've not seen it.
Okay. The flyer it has been posted in our community announcement group. So you just go to the announcement group, you see the flyer. Luckily, by the grace of God, our lead speaker is currently one of us. He presented also, and his presentation was quite wonderful. So when you see the flyer, you will know the person I'm talking about. Let me just keep quiet. And by those grace, we believe that Wednesday will be powerful. Now the way we are planning Wednesday is no much theory, like no much coming to share slide and then you are talking too much. It's purely discussion. Topic will be a little intro and people will share their ideas. And I'm looking forward for us to have arguments on Wednesday, because we have not argued. What we only do here is that we are
Oh yes, member.
We are agreeing. And that is not science. We keep agreeing to what somebody will say. We agree. We all agree. We say, yes, that's good. No. We need to see where somebody will raise the point. I want professor there. I want doctor to say, no. I've done what he just said on this. It is not supposed to be so. No way you are doing scientific argument. Oh my God. I would love to see Professor Adiola participating in such an argument, and that will be.
Ha. It depends. Maybe I'll just agree.
So this is our webinar for February. We'll be talking about predictive modeling in biomedicine, prognosis, and pollution control. Predictive modeling, and our lead speaker is Dr. Olabi Otimi from medical pharmacology and therapeutic, Obafemi Awolowo University, Ilefe. So Wednesday 8 p.m. Kindly share that advert. Let it get to far and near. Yes, keep sharing because we know you have been sharing. So keep sharing. And the advert for 3rd week free training, that is after next week, the next week free training will also be out this weekend. So let's keep preparing for that. Also remember that we are kicking off green chemistry from Tuesday. On Tuesday being 10th, we are kicking off green chemistry. So every necessary information for that shall be released tonight or tomorrow morning, so that we get ourselves prepared for green chemistry. And also prepare that we have a training on March, which is basically our macro 4 modeling, macro 4 design, and all that, all put together for March. Whole will be on that area. I want to say welcome. Okay. Yeah. Good night, everyone.
Good night,
so your
sir.
your certificate will be
Good night,
processed between now and
Good night.
Monday. You have processed the certificate and you all get it. Thank you so much.
Thank you, sir. Looking forward to Wednesday.
Yeah. Thank you.